Hi guys, today we will be discussing our project topic, which is viral gene therapy for Tay-Sachs disease. The viral therapy we will be focusing on is adeno-associated viral vectors. The photo shown here is a photo taken to show the exact position of the chromosome 15 that is mutated in Tay-Sachs disease. Let's start with an overview of Tay-Sachs disease. Tay-Sachs disease is a lysosomal storage disorder caused by a mutation on chromosome 15 gene hex A, which codes for a lysosomal enzyme called beta-hexosaminidase A, but we will refer to it as hex A. Hex A normally breaks down a lipid called GM2 ganglioside, which is mainly found in neurons. The mutation of hex A gene over time leads to the accumulation of GM2 ganglioside and causes progressive damage and destruction of the neurons, resulting in Tay-Sachs symptoms. The symptoms mainly occur in the central nervous system in infants. Things such as muscle weakness, low muscle tone, increased startle response, and sudden contractions of large muscles when falling asleep occur. Sadly, without a cure and severe effects to the CNS, usually the result is death of the infant with a diagnosis of Tay-Sachs. Tay-Sachs is an autosomal recessive disorder, so males and females are equally affected. For example, if you had a mutated hex A gene in both heterozygous parents, you would have one child with the disease. This disease mainly occurs in isolated and interbred populations. The disease is most common among people of the Eastern European Jewish descent.
Tay-Sachs can be diagnosed by determining the activity of hex A through serum, leukocytes, tears, or any body tissue. Genetic testing for the hex A gene mutations and sequencing of the hex A gene can be used for diagnosis as well as heterozygous carrier detection. Infantile and juvenile patients rely on supportive care to manage their symptoms. Their supportive care usually involves neurologists to manage seizures, gastroenterologists, surgeons, and nutritionists to manage their feedings, and lastly, physical therapists to assist with their daily living and mobility. Late-onset patients rely on supportive care, although they deal with slower but more progressive degeneration in these conditions. Enzyme replacement and gene therapy will continue to be researched. Genetic counseling is available for individuals that are at high risk of passing down the disease to help prevent it from happening. Couples at risk of having an affected child have other options like amniocentesis, chronic villus sampling, artificial insemination, ovum donation, and in vitro fertilization. What is AAV? AAV is an adeno-associated virus that is used for gene therapy. Scientists like to call this the amazing virus because it is the only known virus that does not cause disease in humans. AAV contains a single-strand DNA sequence that is easily edited. Splicing is done throughout the AAV to obtain the part of the strand needed for gene delivery. The part that was spliced out is replaced with the new gene, which allows for the AAV vectors to transport the new gene into the patient's body. A helper virus is used to make copies of AAV vectors. This was done by combining both AAV and the helper virus separately with the bacterium, which essentially forms a plasmid. The AAV plasmid and the helper virus plasmid were introduced together in a mammalian cell for replication. The mammalian cell helped produce more AAV vectors. When the AAV vectors go into the human body, receptors on the surface of the virus allow for binding of different things on the cells.
To study the disease mechanisms and to test the gene therapy approach, 12 patients with Tay-Sachs disease were used as models. Our proposal model of study will be reviewed under an IRB extensive questionnaire to ensure safety and fairness. The patients under the trial will have the right to leave the trial at any time, never be forced to do anything that makes them feel uncomfortable, etc. Our participants will greatly be impacting those in the world struggling with Tay-Sachs. We know the fear of a clinical trial lingers, but we plan to have many different professionals around monitoring our patients. To test if individuals have unusual hex genes, their blood was drawn. With a total of 12 individuals, 6 patients with the disease got no treatment and were used as a control, and the other 6 voluntary patients received injections with different types of viral vectors. As shown in the figure, the first condition of this study was injecting 2 patients with the vector that encoded for the alpha subunit of the hex A gene only. The second condition of the study was injecting 2 other patients with the vector that encoded for the beta subunit of hex A only. In the third condition, the last 2 patients were injected with a vector containing both alpha and beta subunits. It is important to recall that mutations in hex A lead to the lack of production of the alpha and beta subunits, leading to Tay-Sachs. Exact injection will occur at the internal capsule of the brain. This area of the brain is associated with motor skills and functions, which are the main impairment of patients with Tay-Sachs. Injection at this site will allow distribution of the altered hex-A gene carried in the AAV in the central nervous system. Now that we've injected our patients with the AAV coding for specific subunits, let's go into the cell and see what's going on. The adeno-associated virus that contains the gene is injected into the patient. The AAV docks onto the host cell using specific receptor proteins and is transferred through the cell in an endosome or vesicle. The adenovirus has components called pentons, which are released from the vesicle once it has passed through the cell membrane. The AAV has specific genetic material that replaces the disrupted gene through splicing with the therapeutic gene. In this case, the therapeutic gene is hex A, hex B, or both, which will restore normal function of the proteins. A protein shell called a capsid surrounds the genetic material and helps delivery to specific areas of the cell, in this case, the nucleus. Once docked onto the nucleus, the capsid is shed and the genetic material forms an episome. An episome is not passed on to future generations of cells, ensuring effectiveness of gene transfer. The specific mechanism of replacement for the DNA in the virus occurs via the virus reaching the nuclear pore, where it delivers the viral DNA, resulting in genetic information from the new gene being transferred through mRNA, creating a new protein. This method was performed using a vector that encodes for the alpha subunit, beta subunit, and a vector that encodes for both subunits. AAVs are ideal carriers because they can affect dividing and non-dividing cells. While the mechanism of adeno-associated viral vectors is extremely complex, this is a good overview of how it works in our patients.
Now let's discuss our results. Shown in this graph, we see a comparison of hex-A activity and GM2 levels. As we can see, the individuals that received the alpha subunit only had slightly more hex-A activity than those that received the beta subunit only. Overall, the patients that received both alpha and beta subunits were showing more hex-A activity and less GM2 levels. Enzyme analysis was performed before and after treatment through the patient's serum to detect the level of protein produced and protein activity of the hex-A gene. As we can see in the graph before treatment, patients had very low hex-A production and the hex-A activity was less than 10%. After the treatment, we can see that the patients had an exponential increase in hex-A production as well as hex-A activity. The patients that showed the most increase in hex-A production and activity were those receiving both alpha and beta subunits.
Using the blood samples from our 6 patients, we also performed a Western blot analysis before and after treatment. The Western blot analysis allowed us to detect whether the specific hex-A gene was being expressed in our patients. The technique allowed us to separate the different proteins that were being isolated through the gel electrophoresis. We then transferred the isolated proteins from the gel to a membrane which was exposed to antibodies specific to the protein. Through the binding of the antibody to the target protein, we were able to determine if the gene was being expressed in the sample or not. In this figure, we can compare the Western blot of a human with normal functional hex-A and hex-B genes with someone who has Tay-Sachs disease. In the normal human, there are 2 bands, one for each gene. The patient with Tay-Sachs disease, however, has no band for hex-A and 1 band for hex-B. The Western blot analysis of our 6 patients before treatment showed no band at hex-A. However, after the treatment, the patients who received both the alpha and beta subunits showed both bands in the Western blot analysis.
As patients started to show recovery of the hex-A gene, they went to occupational and physical therapy to help regain their mobility. The adeno-associated virus will continue to do its job as time goes on. It is only going to get better from here for these patients.
Imagine hearing the diagnosis of your child or loved one of basically the verdict of a genetic death sentence, Tay-Sachs. Although rare, it is fatal. Knowing the child will likely not live up past 5 years old and instead live a life of blindness, seizures, and paralysis. With no cure, there's no hope. Adeno-associated viruses may be the chance that this course could be altered. Although it is far from certain, these engineered viruses may be able to effectively change cells in the brain. We hope this video was informative and possibly sparked interest into further conducting research to hopefully one day come up with a cure to this horrible disease. Thank you!